Good afternoon, everybody. I'll give you guys a couple more minutes as we have more attendees joining, and we'll go ahead and get started. Good afternoon, everyone, and thank you for joining us for our Research and Focus Getting to the Root of SCN webinar this afternoon. We so appreciate having you, and we hope that you learn a lot and take a lot away from this webinar. A few housekeeping items. You do have the opportunity to ask questions throughout the webinar to all of our presenters. You can either insert those questions in the Q&A box below or the chat box, and Eric, our moderator, will get those answered for you at the conclusion of the webinar. Also, at the end of our webinar, we will be giving out a Carhartt cooler backpack with our Farm and Focus logo. We will announce a winner at the end of our webinar. If you are the winner of the Carhartt backpack, please just stay on and we will get your address and shipping information from you to get you that backpack sent over. So we'll go ahead and kick things off this afternoon with our moderator and Director of Agronomy and Research, Eric Osland.
Hey, thanks, Samantha, and welcome everybody. And really excited today to have the two speakers that we do on the topic of SCN. I think our two speakers really understand SCN management and testing and genetics, and really want the participants today to feel free to ask any questions that you have on SCN while we have these two experts online. And just go ahead and put your questions in the chat and we'll get those answered for you. So to start off, we have Dr. Mandy Bish. She is the interim plant pathology team lead at the University of Missouri, and she also is the director of the SCN Diagnostics Lab at the University of Missouri as well. So Mandy's going to start off talking a little bit about SCN management and testing, and just a little bit of an outlook on soybean cyst nematode resistance as well. So Mandy, if you want to go ahead and share your screen and start your presentation, that would be excellent.
Okay, thanks, Eric. Get here. And thank you everyone for joining today. And hopefully you'll share. There we go. Are we good, Eric?
We are. Thanks.
All right, thanks. As Eric said, my name is Mandy Bish, and I'm going to talk a little bit about management. And right up front, I want to give some acknowledgement. So I took over the interim plant pathology responsibilities in April, and some of the latest research that I'm going to share today comes from the people and the funding sources here. So Jeff Bearzone is our senior research specialist in the SCN Diagnostic Clinic. He is great with his nematode identification skills, and he keeps me in line during this transitionary process. Caitlin Bissonette and John Laurie. So Caitlin Bissonette led a lot of the SCN research here before she left the university. And then John Laurie led some, worked with Caitlin on some strip trial data and summarized it. So a lot of what I'm showing today, I've only been here since April. A lot of what I'm showing today comes with the help of these people, and as always, the funding sources who are supporting this effort on SCN. So just a brief background, estimates about $1.2 billion annually, making soybean cyst nematode a very, very problematic pathogen. And even further back, let's take a step in time. So first detected in the US in 1954 in North Carolina. And one of the challenges with this is that the eggs can persist in the soil for years. Okay, so once you have a problem, it's not going away easily. And trying to be proactive on management is one of the best things that we can do. So what did this look like? So initially, back in the 1950s and 60s, when they had SCN present, they had visible symptoms on the soybean plants. And so that kind of led initially for this thought of, if you have a lack of visible symptoms, then you don't have a problem. And as most of us know, and I'm guessing most people on this webinar know, that's not necessarily the case. But I still, even in my short time since April, have had the opportunity to hear from some farmers who are assuming that when they don't have symptoms above ground, then there's not a problem below ground. So in Missouri specifically, SCN was first found in, or first, basically, the nematode lab started because there was this realization we don't understand how problematic this is in Missouri. If we're basing it off, we can't base it off of symptoms, so we need to understand that. So people started sending samples in. It was first found in the Boot Hill region in 1956, and for a little while there was actually a quarantine to try to prevent it from spreading in other parts of Missouri. And over time, you can see how this progressed throughout the state. Whether it spread or whether there was more of an awareness, I'm not sure about that part, but as samples came in, this is where they were confirming the samples from. And then in 1993, all of our soybean production regions, predominantly except for Mercer County, had confirmed SCN presence. And then, Mizzou has had a history of excellent nematologists, and Melissa Mitchum's group did some survey data, and I think Andrew's going to talk about it a little bit or touch on it a little bit later. And, we did find it in Mercer County as well. So you have it across the state. This is pretty reflective of soybeans in the U.S. in general. Where you have soybeans in the U.S. in general, you have SCN presence for the most part. And so one of the things I've been able to do in my short time here with the team, was fortunate to step into a good team. And we did a little bit of spring sampling with funding from SCN Coalition and Missouri Soybeans. So we went to fields throughout the state. The counties are indicated by the yellow stars. And this is just a synopsis of what we found so far. We typically have this threshold of 10,000 eggs as being very high. Anything higher than that is very high numbers. And we did find some fields that had that. We also found some fields where we didn't find any SCN present. And so our goal is to go back to these same fields in the fall, sample again, and see how the management strategies that were used during the season affected the number of eggs in the season. And I like this graph because it comes from Missouri data. So Caitlin Bissonette's team had about 300 samples where they basically were measuring SCN egg counts and then correlating it to soybean yield. And I just broke it down simply for ease. It's a message that has been preached for a long time, but essentially if you have high SCN egg counts, you are going to see lower soybean yield than you do if you have low to no SCN egg counts, okay? And so just regardless of the management practices that are currently being used, if you have SCN at high enough threshold, you are going to see yield losses occur. And so just, the solution, the first one, grow SCN resistant soybean. And so in 1968, the first set of SCN resistant cultivars were released. And, most of our commercially available SCN resistant sources come from PI88, or the resistance comes from these two, PI8788 and the Peking plant introduction lines. And so, that's kind of terminology. Basically, there is a collection of soybean that came from throughout the world, and there's about 16,000, I think, different soybean. There's more than 16,000 different plant introduction lines, so each different soybean. And from all of those 16,000, you have two soybean where we have most of the resistance from. And Andrew can take you into much more detail on that later. But for our purpose, for management right now, what we need to know is that greater than 95% of our SCN resistant soybean grown in Missouri use one source of resistance, the PI88788. And so, we learn from herbicide resistant weeds, for example, if you use the same source of resistance over and over and over again, then the pathogen or the pest is going to develop resistance to that mode of action. And so that's currently what we're seeing right now. Melissa's group did some surveying where they actually Dr. Mitchum went out onto Missouri soils and collected samples. So different than samples being sent in, they went out and they collected. And what they found was that SCN populations that they collected, 100% of the samples that they found where they found SCN present, those could actually reproduce to an extent on this PI 88788. So what that tells us is PI 88788, which is in your soybean, is providing some protection against SCN, but we're losing ground on that protection. Okay. And so what are some other tools that we can use to stack our protection against SCN in addition to those resistant lines? And so seed protectant treatments have been on the market for a little while now, and Dr. Bissonnette and Jeff Barazone have some data in greenhouse setting where they've done some extensive analysis on different seed treatment options against SCN. And so one of the things that they looked at in this study was reproduction and looking for the females and doing female counts on soybean plants, so sensitive soybean plants that had the seed treatments on them. And so the SCN population type I'm going to show you is 2.57. And really, Andrew may go into more detail on this also. Sorry, Andrew, I'm pinning a lot on you here. But what this means to you is that the SCN populations that were used in this study could reproduce on that PI 88788. And so that's why I pointed out here. So we know that we're already losing some ground with this soybean line that's being used. And so in the greenhouse, they saw very consistent effects on SCN numbers. And so this is a busy graph, but if you look on the right, you have a non-treated soybean that has that is susceptible to SCN. So it does not have the PI 88788 in it. And this is the number of SCN females that they found on that soybean 30 days after planting. The two colors are different years, so 2020 and 2021. So then if you look at the next set, this is the non-treated, so there was no seed treatment, but it is a variety that has the PI 88788. So as you would expect, you see less females present than on the susceptible. Then we have our base treatment, which is does not include any of the nematode protectants. And you can see that in 1 year, they did see a reduction in SCN females, but not in the other. But what I really want to point out, now that we have these controls over here, is that Alivo consistently, regardless of the SCN population tested, consistently had a reduction in the number of females. And you see some reductions in the other treatments across the years. So the black corresponding to this basic seed treatment here, you can see the reductions. It's not my job to promote one product, definitely not my job, but they did see the most consistent results with Alivo across the greenhouse. So, it is great to see that, but how does that translate into the field? And so Dr. Bissonnette led a multi-state and multi-country project to look at the seed treatments in the field. And I just have some punchlines here of what they found. So this is not published yet, but there was less consistent suppression of SCN reproduction when you had those seed treatments out in the field. Sometimes Alivo worked to suppress SCN, sometimes it didn't. There was, though, in all the treatments, a yield increase above the non-treated seed. So there was a benefit to having the seed treatment, but we weren't seeing the reduction necessarily in SCN. And across, if you take all the environments and all the conditions, all the different population densities, whether you had SDS or you didn't have SDS, Alivo and Saltra had higher yields than the seeds with the base treatment. So those were the two treatments that really had the higher yields, and those are our chemical protectants. So that's what that looked like. Now in Missouri specifically, the Missouri strip trial program did a test with Alivo specifically. So on farm research, on farmer fields, they took strips of soybean that had treatments compared to strips that did not have Alivo seed treatment, okay? And this is a very busy graph, and I'll try to walk you through it briefly, but it illustrates the point nicely. So each row represents one location where the study was conducted, okay? So each row is one location. The green diamond represents the average of all the strips, the yield of all the strips that had Alevo treatment. Okay, so each individual yellow was the yield from an Alevo treated strip at that location. Each black circle was the non-treated control. Okay, so you can see that at this top site, there was about a 9 bushel per acre difference between the Alevo treated strips compared to the non-treated strips. Okay, so that's how you read this. That's one location. When you take all of this data together, you get about a 2 bushel per acre average increase in the strips that had Alevo compared to those that did not. Now, we know that SDS, there is some linkage between SDS and SCN. And so the locations that have stars by them, those were locations that had an SDS pressure that was observable. So you could see the SDS in those strips. And so if you just take those sites specifically, the average difference between the Alevo treated strips and the non-treated strips was 3.5 bushel per acre. Okay. And one of the reasons I wanted to share this data is because it's Missouri specific and on farm. Another reason is, you know, we're always looking for more opportunities to do more on-farm testing. So if there is a farmer out there that you know that is interested in seed treatments, it's not specific to Alevo. At the time the study was done, that was the main product out there, but we'd be open to hearing from people who wanted to do some more on-farm research on this. Okay, so seed treatments, the punchline is they can be effective. They're not always effective. There is some environmental work at play, and I'm hopeful, you know, that as my time as interim director continues, that I can dig in. One of my strengths is environmental analysis, so bringing environmental analysis to this data and trying to tease apart why we see a difference in some fields versus others. Okay, so one thing that you do have control over, you don't have control over the environment, but one thing you do have control over is something that we know, but we rarely take time to do. You know, nematodes can only move inches, right? Max, very small distances by themselves. We are the ones, and water are capable of moving them further. So if you have an area in your field where you have seen, maybe it's lower yielding in general, just this area that you can't explain, or maybe you see SDS show up commonly in that area, you're going to want to run equipment through that area last. And I know it's easy for me to say that. It's much harder to carry out in the real world. I appreciate that. But really, this is one of those pests that once it's there, it's very difficult to get rid of. So minimizing the spread, doing what you can do is important. And this is one area that we do have control over when dealing with this pathogen. Another thing that sometimes that we don't do as well as we should maybe is rotate to non-host crops. So corn is a non-host crop. Just in our brief survey, there's a lot of work that's done on this in the past, but just in Missouri in this year, when we did that spring survey, we just divided it up. So the 2021 crop, you can see, was corn or soybean. And these are the egg numbers from each of those fields. Just on average, there was maybe 2.5 times less eggs after corn than there was after soybean. Okay, so you know, the eggs can persist, the cysts that hold the eggs can persist in the soil for years. So it's not like 1 year of crop rotation is going to get you out of it if you have a problem, but it is going to help decrease those numbers, and that's another thing that is within your control. So, and finally, cover crops and SCN. So there's some work that has been done on this. There's a lot of work left to do on this. And I just wanted to share some work that came out of Dr. Tilke's lab up at Iowa State recently. He's been pretty outspoken about cover crust as a potential host for SCN. And I don't know if Andrew's going to talk a little bit about that or not, but I wanted to look at some of these grass cover crops that we use in Missouri, because I know there is the SCN Coalition has been promoting annual ryegrass, which is a non-host, and I understand that. But if I try to promote annual ryegrass by itself, Dr. Bradley, our state weed scientist, is going to come down and say, what are you doing? Because it is very easy for annual ryegrass to escape herbicides and become weedy really quickly. And so I just wanted to give you an overview of a project that Dr. Tilke's group did recently, looking at different cover crops. And this was in a greenhouse setting. And they basically planted cover, they did several different studies. On this one, they planted cover crops in pots. Then they took the pots outside over the winter to let them winter kill. And then they came back and they planted a sensitive soybean in those pots. So the initial pots, before they planted the cover crop, they had a known amount of SCN present in those pots, okay? And so then they were like, okay, well, how do these cover crops and their, after their winter killed, how do they affect the levels of SCN? And so again, a lot of bars on this graph, but if you look at the far right, soybean, so this is your sensitive soybean. So you had sensitive soybean planted, you didn't have a cover crop, and then you came back and planted sensitive soybean, and you can see the number of soybean cyst nematode females per soybean right here. So we're really comparing to this number. And what we see, we know tomato is a non-host. Anything that has asterisks above it, we're significant reduction in the number of SCN females compared to the soybean alone. And so you see cereal rye and the annual ryegrasses really do, they at least don't promote SCN female development or reproduction, I'll say that. I don't know if we can call it reduced yet in a greenhouse setting, but they don't promote it. And so they have potential as non-host winter crops in your rotation. And so, the current recommendations are kind of plant a non-host crop like corn, plant a grass cover crop, and then you can come back with your soybean. Finally, another thing that you can keep, that you have control over, is just keeping up to date on SCN resistant soybean varieties. So, when I look at options with peaking resistance for maturity groups in Missouri, I haven't seen a lot of maturity groups. I actually haven't seen any, but if I would love to be correct if that's wrong, but I haven't seen any that are above 3.2 yet. And so that's going to work maybe for our northern producers, but for our southern producers, that doesn't leave a lot of options. And so what I want to, say about this is that one thing to consider is that most of your commercial soybean varieties have that PI 88788 that I talked about, but there are different levels at the molecular level of that. And so rotating among your commercial varieties that have that PI 88788 can offer a little bit of help too, along with the seed treatments and the non-host crops. And so finally, knowledge is very helpful in making management decisions on SCN. And I kind of like this iceberg. Sometimes I think it's used out of context in posters, but what is happening under the surface that we don't see? So, you have control over knowing what the SCN pressure is in your field. And with support from Missouri Soybean and also from the SCN Coalition, we can provide some free samples of farmer fields for fall SCN egg counts. And so the recommendation I have here is, it can be fairly frustrating to sample for SCN eggs if we're being honest, because it can vary so much across the field. So getting enough samples, taking enough soil samples is key when it comes to knowing your numbers. So the recommendation from the SCN Coalition is you want to take 15 to 20 soil cores, 1 inch in diameter and 8 inch deep. So a little different than other soil sampling methodologies. And you want to combine those and do that for every 20 acres. Okay. And so right now we can offer, between Missouri Soybean and the SCN Coalition, we can offer 4 free samples per farmer. So you can contact me if you're interested in that, please do. And then the other thing is that has been promoted lately is female counts in season. And so that's where you dig up the roots and you look for the females, which are white, but a lot smaller than nodules. One of the keys for this though is you have to dig the roots and you don't want to pull the roots. Now, in the literature, the suggestion is that egg counts are more representative of end of the season yield, but it can be easier to see the females on the roots. So there is some benefit in that as well. The one thing I will say about the females is, I have this red arrow here to remind me to say it. The females are attached to soybean root by their mouth, so they will come off easily. So when you're digging up these roots, you have to be pretty gentle when you're cleaning them off, to look for females. And also we can do those in the lab. We're set up to do that in the lab as well. And so with that, I just want to leave you with a couple of resources. So the knowledge, what's your number? You have control over that part. And the SCN Coalition has a website, and SCN Diagnostics is the MU lab for the testing. And I'll be happy to take questions at the end, and I'll turn it over to Andrew now.
All right, thank you, Mandy. Very good presentation. And yeah, just for the sake of time, we will save questions for the end, but remember that if you do have any questions, to put those in the chat, and we will get those to the correct presenter and get those answered. Next up, we have Dr. Andrew Skaboo, who is the soybean breeder at the University of Missouri for the Northern Soybean Breeding Program. Dr. Skaboo manages several projects, fairly large, well-known projects, as a matter of fact, on soybean cyst nematode, and has really kind of built his program to be one of the top programs in the nation as far as soybean cyst nematode genetics goes. And really excited to have Dr. Skaboo on this call today to talk just a little bit about what is the genetic status of soybean cyst nematode resistance in soybeans, and any other information that he wants to share. So Andrew, feel free to take it away.
Hey, thanks a lot, Eric. Can you guys hear me okay?
We can, yeah.
Can you see my full screen okay?
Yes, you're good.
And my pointer?
All good.
Okay, good. Well, I think Eric's right, except for that the University of Missouri as a whole has really led for 40 years SCN research, starting with, well, not even started, just a plethora, Prakash, Grover, Henry, Melissa, Caitlin. We've just had a real strong SCN program here at the University of Missouri for a long time. And I think many may say the same thing. We're just trying to continue on that great tradition of world-class research in soybean cyst nematode here at the University of Missouri. So how we've contributed over the last few years. I'm definitely like Mandy too, and I didn't mean we didn't collaborate on that, but I like to shout out the acknowledgements first. I want to first highlight Clinton Meinhardt. So Clinton has been with the University of Missouri for over 15 years, and he manages our soybean cyst nematode screening facility. And it's one of the best in the world. And big reason for that is Clinton's work. I also want to highlight here Paulin Bassinet, Dr. Paulin Bassinet. Dr. Bassinet recently graduated. I think I may have even seen him on the participants today. But a lot of the research, genetic research that I'm going to talk about today was part of Dr. Bassinet's dissertation program and recent publication. Then I'd also like to highlight Dr. Mariola Plodno. Usovsky, sorry, she recently married. Dr. Mariola Usovsky is also an expert in soybean cyst nematode genetics, as well as reniform genetics, root knot nematode genetics. And Mariola leads our molecular biology lab here at the University of Missouri. So, I think Mandy did a pretty good job of how important soybean cyst nematodes are. And I would just highlight, for me as a soybean breeder, it's one of those biotic stresses that really puzzles me if I was a farmer, because like Mandy said, you don't see it. You can go out and see and know you have SDS and know you have frog eye coming in, or you can see Phytophthora, Pythium damping off early in the season. Nematodes, when you can potentially have yield effects on your yield, detrimental effects on yield due to SCN, you're largely not going to see that in foliar symptomology. So what Mandy and the SCN coalition talk about with knowing your number, it's just a really important point for farmers, and something I hope we get across today. Just know what's out there. Take a test. It's free. The University of Missouri, Mandy will run and tell you exactly if you have a real bad nematode problem or not. Yeah. And a little graphic on the distribution. And again, Mandy's right. A lot of this is done from soil moving around, not the nematodes moving around in the soil. But it's all over the Midwest. And what I want to highlight, I'll start here. So we talk about two different, and it can be confusing, I think, for farmers, but traditionally we've talked about races when it comes to soybean cyst nematode as a pathogen. And nowadays, we also talk about, especially in the academic setting, we use HG type. And there's a difference between the two, but both systems are a way to understand the population and the pathogen that is in your field. And the biotype that is in your field. And what I want to highlight today for everyone on the call is how important that concept is. Not only knowing the number of eggs, but knowing how virulent is that population in my field. And that's done through the SCN diagnostics program by just looking at what biotype. And just a real quick example. So we have the HG type test, which is what SCN diagnostics does. And then we have 7 different indicator lines here. And these are all specific soybean germplasm that has a known and well-established reaction to soybean cyst nematode, whether it's susceptibility or resistance. So what I want to point out here is that our number 2 indicator line is this 88788 line. And I believe Mandy mentioned this, but that is indicated when you see HG type 2 in the biotype assay. That means that population of soybean cyst nematode is able to reproduce on 88788. And that's a really important concept for moving forward with what I want to talk about in the research that we're moving forward with. Okay, and that leads me to the types of, you guys know this, we've been talking about soybean cyst nematode a long time. I don't know, I was born in the 70s, right? We were talking about soybean cyst nematode in the 70s. And traditionally, I'm sure all of you have heard that we use this 88788 type source. And what I want to make sure everyone understands is I'm going to use the gene terminology during this presentation. And if there's any questions during this, Eric, stop me. And, please, I think the easiest way maybe we're just to log your question into the chat. And then Eric, you can stop me and ask. But it's an important concept because we talk about genes and genetics. Genes are what controls resistance in these plants. And I'm sure everyone can, I'm hoping that I can explain this well enough so everyone can follow it. So our traditional 88788 type source, this is a germplasm line in the USDA germplasm collection that harbors this gene. We call that RHG1B. This gene is responsible for imparting resistance to race 3 and 14. And I really want to make sure everyone understands that. RHG1B, the gene that's found in 88788 that is bred into commercial cultivars, is responsible for controlling to resistance to race 3 and 14. Okay, so then we have our popular Peking type source. And the difference is that Peking has what we term RHG1A. So RHG1, it's the same gene in the same place, in the same chromosome, on the same chromosome, but it is a different allele. And we could talk about that, but that's not as if, so we have 2 different alleles. Peking type source has a different allele or an alternate form of the gene RHG1. Okay. To have Peking type resistance, you also, in addition to RHG1A, you have to have this RHG4 locus. So it takes two genes, and it's what we term an epistatic interaction between these two genes. That means that one gene by itself does not confer resistance. If you only have RHG1A, you will not have resistance. If you only have RHG4, you will not have resistance. You have to have these two genes together, the resistant allele of those, to impart resistance. I want to highlight again, this is for race 3 type soybeans, and we use either HG0 to determine that HG type. Okay, so I want to ask the genetic line. We have one gene in 88788 that controls our resistance. In Peking type, we have two genes that control resistance. Okay, does that make sense for everyone? If there's any questions, please send them to Eric and we can. One more thing I want to highlight is this is our traditional understanding of Peking type resistance for the last 30 years. This is our basic understanding of Peking type resistance and 8.8 type resistance and what the breeders in the industry have used. But I want to highlight that this has really been focused on race 3 type resistance. Okay, so there's a lot of different strategies for SCN management. I think Mandy did a great job of letting everyone know those different strategies. Seed treatments, crop rotation, and resistant cultivars are really how we're going to tackle this pathogen, but they all have their own, limitations. Seed treatments seem to not be as effective all season long. Crop rotations only really reduce inoculum. We're not having as much of a shift, and crop rotation won't help you with the spread of more virulent nematodes, and resistant cultivars can break down. So we got, we're a team. We got to face this problem as a team because they all, each by itself, all has issues. So I'm going to highlight a survey done by Melissa Mitchum's group for Missouri, and I want to highlight why this is so important. So if you remember, if you go back to our HG type test, well, let me first start. I think this is the exact same map, but just a little different information presented as Mandy presented. It's just showing where SCN is detected all across Missouri. This was published in 2016. But what I want to highlight here is, yes, SCN was detected everywhere, but look at our HG types for every area. They're all either 1, 2, or 2 by itself. And if you remember what we started with, if you have a HG type name with 2 in the name, that means that it can reproduce on 88788 type or RHG1B type resistance. Okay, and that is every single county in Missouri. So, farmers, people out here, audience members, I think a real argument can be made that we currently do not have sufficient SCN resistance in modern soybean cultivars. I mean, it's getting to that point where it's not anecdotal evidence anymore. And I'll back that up with just a little bit of data here in a second. But I believe, and this is also what, or I think our next slide, yeah. This is what Mandy mentioned. So in every single region in Missouri, all of the nematodes that Dr. Mitchum's group and SCN Diagnostics collected, 100% of them were able to reproduce on 88788. So if you had that one gene. So that's a problem. It's a real big problem. For audience members who are interested, there are 2 really good papers recently that came out, and they're review papers on all of and it's really twofold. One was done by Dr. Andrew Bent at the University of Wisconsin. The other one was done by Melissa Mitchum at the University of Georgia. And these 2 papers together explain where we are in science from the plant genetic resistance side, that's Dr. Bent's review, and then from the nematode genetics side, and that's Dr. Mitchum's review. So together, I really encourage anyone who hasn't read these to read them, and you can really get caught up pretty quick on the current state. So I'm going to highlight, again, Dr. Besnet's research that was cited in this recent review. And we published this a few months ago in Theoretical and Applied Genetics. And well, let me just say the take-home message to start and end with. The take-home message is that what we found is that Peking type resistance does not include only 2 genes. It's actually Peking type resistance. True Peking type resistance is the result of 3 genes. And it's how 2 genes interact with RHG1 in epistatic fashion that gives you that resistance, multiple resistance to multiple virulent and non-virulent nematodes that is found in the cultivar Peking. And this was, this was a pretty big deal. We had a lot of interaction with this because it explains a lot of what's been going on. So we, I might move through this a little quickly, and I'm not sure how I'm doing on time. Just let me know, Eric. But we did QTL mapping in this work, QTL mapping and fine mapping. We developed populations. We've taken the approach beyond, this isn't 30 years ago. We have a lot of information on nematode. So we're just not blindly taking a resistant variety and crossing it to a susceptible variety, right? We're being a little more pragmatic and a little more deliberate in our population development strategies for mapping techniques to get at to answer the questions that we want to answer. And honestly, the first question was, what can I stack with RHG1B from the really resistant line and gain resistance, better resistance than what I have with RHG1B? And we answered that, and the question is nothing. Or that we tried to answer that question, and the answer was nothing. We found that you cannot stack or pyramid any genes with RHG1B and fight the most virulent nematodes that are present. So we developed these unique populations. We did the genotyping, the phenotyping. This is a picture of Dr. Besnet. Dr. Besnet. I'm going to say, I just wanted to highlight here with what the program that Mandy leads in our breeding program. We have one of the best facilities in the world and the best protocols and staff in the world to do this type of research. And it's a big deal. So just getting to the results real quick. What we found was when we looked at, when we make a marker, so what you're looking at here is a significance level on our y-axis, and then all the 20 chromosomes in soybean on our x-axis. And what we found was on chromosome 18 and chromosome 11, we had, because it was above this orange line, a significant correlation between that genomic region and our phenotypic data. And what that means is that there's likely genes in that region that's correlated that control our trait of interest. It's QTL mapping at its simplest form. So we found that these 2 genes. What I want to point out is, hopefully, if I did a decent job of explaining, you do not see RHG4 here, right? So traditionally, Peking type resistance is known to be controlled by RHG1A and RHG4. We did not find that RHG4 was relevant to resistance in soybean cyst nematode. We found that the major driver was the interaction between RHG2 and RHG1A. And why is this? That is because we used a virulent nematode population for our mapping, okay? I can't stress that enough. So traditionally, all the mapping, much of the mapping for the last 30 years has been done with HG type 0 or a race 3 14. Again, that's irrelevant. It's really, I don't think it's almost, I think it's easy to say that is irrelevant when that population is not found in Missouri, right? These are the types of virulent nematodes that are found in Missouri. RHG4 has nothing to do with controlling that type resistance. So that's the first major finding that we had. And not that this is a new finding, but the interaction, how these genes interact when they are pyramid is brand new, right? And it's not additive type action, it is epistatic. And we confirmed this with a different type of analysis called a nested association mapping analysis. As you can see, same chromosomes, same genes were found, correlated, same regions correlated. Then we took the next step in genetics work and we find mapped this QTL, meaning if you think of a Google Earth, when you think of a chromosome, maybe you're looking at Missouri. So we narrowed it down to this little region where, oh, well, now we're on Hit Street, right? As far as where genes are, right? So we're really narrowing down the region of where of the region of the chromosome that is responsible for our trait. And what we found about only 21 candidate genes. So we really mapped it really small, and we found that the SNAP gene, which is a paralog of the RHG1 locus, was right in the middle of our QTL interval. So really, we haven't cloned the gene, but really, really good evidence that these SNAP genes are responsible. So Eric, can I ask how much time do I have left?
7 minutes.
Okay, good. So I want to start, and this is really the heart of this is the data. These are the facts, the data, hypothesis testing that backs up what this is. So I'm going to start with this first slide, and this is these are results from a population of nematodes that is considered a race 3 type. And this is, again, this is what all the traditional nematode genetic work has focused on, this type of race, because 20 years ago it was the most prevalent, right? Because we've deployed RHG1B so much, it's not anymore. So if we look at this old race, right, this irrelevant soybean cyst nematode population, yes, okay. So this is our race data. Here on the y-axis, we have our female index. So generally anything below 30% female index is what we consider genetically resistant to nematodes, okay? Then on the x-axis I have here is my allelic combinations. What I want to point out is that all of the first 4 combinations include RHG1A, right? This is the Peking type of RHG1 allele. All of the last 4 left to right include RHG1B type resistance, right? So yes, looking at this data, RHG1B controls resistance for race 3 unequivocally. And it really, here is RHG1B by itself, right? If you have only a RHG1B, look at how resistant you have, right? High resistance. Problem is, this nematode is nowhere to be found in Missouri anymore, right? The other important thing to point out here is that our stacking with RHG1A and RHG2 does not control resistance to race 3, right? So our old nematodes, RHG2 has nothing to do with that, right? I mean, you see a little drop, you know, probably not significant. All right, well, it is significant, but you know, we're not getting resistance from it. It's this combination of RHG1A and RHG4 that gives you resistance. And that's why we had that knowledge based on Peking, right? And we didn't think about RHG2 as geneticists, as breeders, because it has no effect on this race, this type of nematode population. RHG1A and 4, that's where you get your resistance to race 3. But race 3 is nowhere, right? What's relevant? These other races, these more virulent races. So now let's look at this. Here's our RHG1B 887 stacked with a whole bunch of different type of alleles, right? RHG2, RHG4, R4. Yes, you can get significant differences, but nothing that drops you below really 60%. So you're always susceptible. If you have RHG1B allele in your germplasm, you are susceptible to race 257. And what drives that resistance? It's this combination of RHG2 and RHG1A, right? It's not this resistance between RHG1A and RHG4, because as you can see, it explain all of the resistance, those two alleles, and many of them you're still susceptible. The majority of the lines are still susceptible to the most virulent races when you have only RHG1A. It is RHG2 that is controlling the resistance to the most virulent nematodes, right? So that's for race 1, same HG type, but race 5, difference is actually determined by picket line. We won't get into that, but that is, but again, we see this, it's the same HG type, so we have the same pattern. RHG1B, 88788 type resistance, you have no control over virulent nematodes. It's only when you incorporate RHG2. And it's this exact same pattern. And I can tell you, getting this high quality of data and repeatable data is really a result of Paul and Mariola and Clinton. So then lastly, here's the most dramatic, right? And here's our most virulent nematode as HG type. Right now, we're up to 4 different indicator lines that this group of nematodes, this population of nematodes can reproduce above 10% on, right? So the most virulent nematode, race 2, that's found in Missouri. Now look at this. RHG1B has absolutely nothing to do with it. And then the combination of RHG1A and RHG4 has nothing to do with resistance. It's all about RHG2, having RHG2 and the epistatic interaction. Remember, epistasis is determined by no effect alone, only in effect when we're together in the genome. And alone, RHG1A does not have an effect. The parent had RHG2, no effect. Only when you bring these two together and you get that epistatic interaction and you get resistance to the most virulent nematodes. And that's what we have out there. Okay, so I'm probably getting maybe I'll just stop there if I'm getting pretty close. That's kind of our big takeaway. So, one big takeaway as farmers, as you ask for, if this tells you, well, let me get some Peking type resistance in my field, I would be certain from your seed rep that it contains RHG2, right? That is because just saying it has, if you say it has Peking type resistance, Peking has RHG2. That's why true Peking type resistance is a 3-gene model, not a 2-gene model. It's only a 2-gene model if you only look at one race of nematodes. If you look at why Peking is such a great variety, why we say Peking type resistance is because Peking is resistant to all of these, right? Because it has all 3 genes. Okay, I'll stop there. I could ramble on, but I'll stop there. And I do want to send one shout out again to Paulin. I think, yeah, he's still on there. That was Paulin's research and works being so good. Thanks. I'll stop there. Happy to take any questions.
Really appreciate it. Yeah, please put your questions in the chat if you have any. Andrew, one question I had kind of while watching your presentation. So do you think that the reason the nematode has kind of shifted, has it shifted to the race 2 from the race 3? Is the reason for that kind of due to selection pressure and the fact that the 88788 and the Peking were so good at controlling race 3 that we've kind of, I guess, selected that race out of the general population of nematodes, and now we're left with a nematode that's more adapted to these other resistance types that we're using, like the 88788 and the Peking without the RHG2?
Yeah, that is somewhat of a loaded question, and I'll preface this by saying I'm a simpleton plant breeder, not a nematologist. And the reason I say that, Eric, is, and it's such a good question, is it's really, when you think of a qualitative trait like high oleic acid, breeding for it, it's pretty straightforward. You have a non-functional mutant, it's high oleic. Nematode resistance, breeding, and genetic resistance you have to think about the nematodes on the population genetics level, where it's really the frequency of alleles and the frequency of individuals within this nematode population that's driving the phenotypes that we see, right? So to answer your question, I think it's yes, right? If you have a nematode population that is constantly subjected to a resistant source that controls a specific type of resistance because of the genetic mechanism in the nematode, then you're going to have simple population genetics theory, just like in weed management, I'm sure you know, where you have a shift in allelic frequency towards virulence genes. Those virulence genes are what we see in the nematodes that are able to reproduce on 88788. So it's not, in my opinion, it's not that like the nematodes are changing, it's that the genetic, the allelic and genetic frequency within the population is changing just due to selection. So, you know, it's and at this point it's becoming unarguable that it's not happening. What we still haven't seen, right, is how much is this affecting yield in farmers' field? How much is it preventing yield gains, right? And I think Mandy showed some of that, right? Like, it's just, that is maybe not as clear, but last thing I'll say, I think there's a reason some of these Peking type that have all three genes are at the top of the test nowadays. I'll let Mandy comment if she wants to say anything else.
Now, I would say that on-farm yield data is a little bit elusive, and just guess I give another shout out to the strip trials, because I really think that's the place where we're going to have the opportunity to figure those answers out. So, yeah.
Yeah, Andrew, I think what you just said about, the varieties that have the peaking resistance with all three of those genes coming out on top of some of those trials really says a lot for what may be happening out there as far as yield loss from soybean cyst nematode, that you aren't really seeing symptoms, but how much yield loss is actually occurring. So
yeah, it's an anecdotal place to start some of these larger project questions that could really hone in on that, and answer that if we're really seeing this dramatic yield difference. Because like I said, I don't think it's inappropriate to say we don't have good nematode resistance in modern soybean in North America. It's, we just don't.
Right. Yeah, that's that was really good. Really appreciate both of our presenters today. I don't have any questions in the chat, but I know that those who participated, like me, learned a lot. And once again, thank you to Andrew and Mandy. We do have a winner for our Carhartt backpack cooler, and that was Cecil. I don't know if that's Cecil DeMont, but congratulations, Cecil. If you want to hang on the Zoom once everybody else hops off, we will get your information and get that cooler sent to you. But once again, thanks everybody for attending. Hope you enjoyed your.
Eric,
yes.
There's a couple questions in the Q&A that I was just going to go through.
Okay.
Yeah, sorry. I was going to type them, but we're running out of time. So
nope. We can definitely go ahead, and they popped in. So,
yeah.
Mandy.
Yeah.
What is the best time of year for sampling, and how often should a field be sampled? And after that, are there field characteristics that impact nematode pressure?
Sure. So we recommend sampling in the fall because that is when you've had multiple life cycles. The temperature warms up in the spring, the eggs begin to hatch, and then you've had multiple life cycles by the fall when harvest occurs. If you are concerned about it, another line of thinking is sample in the spring, kind of like I mentioned, sample in the spring and then come back in the fall and see how the numbers have varied. And that will give you more information on whether your management practice is working. And then, yes, there are field characteristics that are going to come into this, nematodes like sandier soil. And I'm still learning how to piece some of these other environmental factors into it. So hopefully stay tuned for that one.
You want me to answer?
Andrew, yeah, if you want to answer
Yeah.
the other one.
Yeah, so the
about
question came in if our northern soybean breeding program has any soybean lines that are showing promise to SCN resistance. Well, the first thing I'll say is that we don't release anything that does not have some type of SCN resistance. So all of our material that goes through the release process, we just feel like it has to have SCN resistance. Traditionally, that has been RHG1B type resistance. So what I talked about today, as far as true Peking type, 3-gene Peking type resistance, we hope to have at least one variety released in the next 1 to 2 years. We hope to next year. We'll see how the data plays out this year. And then the third way I'll answer that is, this is such a big deal to me, and maybe it's just so fun. But well, it's fun research to do as a scientist, but I think it's really, really important for the industry. And so, our program made the real conscious decision to only work with this 3-gene combo now and in the future for the rest of my career. It's such a, again, I think for a public breeding program, it's good to have that, a public breeding program that focuses on variety development. Do some variety development of novel material that's not on the market, right? That's where we can fit in. If a farmer, if you guys want to go get RHG1B 887 type resistance, there's a lot of varieties out there, right? Well, why don't I focus on a program that has SCN resistance that's unique to the marketplace, to 95% of the marketplace? So I look, honestly, in the future, 5 years from now, we will only release varieties that have this 3-gene combo, or we're working on some new things too, right? But this novel resistance, because again, if you say you just have RHG1B, you don't really have strong SCN resistance, So, yeah, I'll stop there.
Nope. Thank you, Andrew. Mandy, the other question, did you answer that?
Yes.
Okay.
I did.
Perfect. All righty. Well, once again, thanks everybody, and please stay tuned for the remaining webinar series. But have a good rest of the afternoon.
Yeah, thanks everybody.
Hey, Eric.
Yes.
Hey, Cecil, could you stay on for 1 minute? Can you hear us?
Looks like he's still on. Yep, there we go.
Hey, Cecil, if you would, you're the winner of our giveaway. If you could either share with us your shipping information or send me an email at communications@mosoy.org, we will get you your backpack mailed over. Okay, wonderful. I will just follow up with Cecil after our Zoom meeting, and we will get that figured out. Thanks everyone for joining. We appreciate it.